Who among the Lyme disease criminal, CDC and the ALDF staff, have published that OspA, or the fungal osps or triacyl-lipoproteins of Borrelia, could never have been vaccines? So I'm going to give you some background first. Who are we talking about when we say they falsified the testing in order to falsify the vaccine's outcome? It's the ALDF.com. And take a look at these people. It's most of them, not all of them. And the ones that were on this, belong to this organization previously, like Barbara Johnson. And these are the people that we're dealing with. It's nice that they put all their names on this one document, which is very accusatory towards people who say they're criminals when they are criminals, and they've been sued by Senator Blumenthal for antitrust. That means conspiracy. So here's the racketeering complaint I filed with the Justice Department in 2003 upon the recommendation of Senator Blumenthal's 3 staff lawyers when he was the Connecticut Attorney General. And as you can see very clearly, on the left is the neuroborreliosis, or the patients that have the more devastating form of the disease. They're not making any antibodies. They're immune suppressed because the way that Borrelia or fungal antigens cause disease is to cause immunosuppression and then the reactivation of dormant viruses and tolerance to other infections they would have been able to fight off. And those infections do in fact cause disease. And this other graphic is the last page of that RICO complaint. I put it in here for people who might be able to better understand what we're talking about. That the HLA-linked arthritis is an autoimmune disease, and it's characterized by too many antibodies or too many autoreactive T cells against knee joints, or whatever the mechanism is. It's very different serologically from the neuro-Lyme, and they left the neuro-Lyme out of the case definition because it was caused by the OspA, the fungal endotoxin OspA 2. So they changed the case definition in 1994. But previous to that, they said that the way to recognize Lyme disease was to look for new and expanding IgM antibodies, because that meant the bug was still alive. And Alan Steer said that you can use band 41 only, and that it was the earliest band to show up, and that you could use that because you could distinguish syphilis from borreliosis. And then later, after the bogus vaccine trials and the bogus Dearborn stunt, and right before the vaccine was going to be put on the market in early '98, the bad guys, Ron and Evans at Yale, published this book, Lyme Disease Key Diseases Series, and I recommend everybody get it because it's just chock full of scandalous, libelous statements. But in it, this section by Steer, he says, I'm going to quote it, "It should be stressed that seronegative Lyme disease is a contradiction in terms if a patient has marked arthritis, particularly chronic arthritis, because the immune response seems to be involved in the pathogenesis of joint inflammation. Seronegative Lyme is a subtle, attenuated illness." Well, it's only subtle and attenuated in antibody production. It's an immunosuppression disease. So I just want to make sure you know that the bad guys claim you can't have Lyme disease unless it's a bad knee only. And in this same book is Lenny Siegel and his Munchausen's accusations, which you should take a minute to read. Stop the tape if you like, because I have a lot of material to go through, if you want to read what he said. Now, Lyndon Hu is one of the authors, with Mark Klempner, of the ridiculous source of the Infectious Diseases Society of America guidelines on Lyme disease, and they use the Dearborn case definition as the inclusion and exclusion criteria, and that's invalid. So it's remarkable that Lyndon Hu now has changed sides, so to speak, not publicly anyway, but has said that has asked for a grant based on the fact that Borrelia antigens cause immunosuppression tolerance and post-sepsis syndrome, and you're going to see that later. That's the reason I decided to make a video. And here Mark Klempner says, patients generally feel well aside from their arthritis symptoms. So here again, Lyme disease, quote, end quote, the case definition was changed in 1994 in Dearborn, Michigan at a conference the CDC held, and you can only have a case of Lyme if you have only a bad knee and lots of antibodies. I'm including this transcript from the PBS Life on Earth series from quoting Eugene Shapiro, because he's also saying Um, somehow, this is from 2001 or so, somehow that form of the disease, antibiotics are effective. They do fine. But then there's some other form of the disease which you can't put your hand around it. They don't have objective findings of inflammation, which is the way bacteria cause disease. False. We know that they can cause disease by immunosuppression. Look at tuberculosis, for example, and all the other cases. But anyway, I just wanted to give you their mindset. They insist you don't have Lyme unless you're not sick and only have a bad knee. And by the way, Eugene Shapiro's undergraduate degree is in English literature, so he's really not qualified to be speaking on these matters. This next article is by Alan Steer in 1994. I guess it was submitted in 93. But anyway, this is the source of the scientific fraud that went into the case definition of Lyme, where OspA and B are left out of the case definition, and that was to later facilitate Lymerix, as you'll see. You never test for a vaccine's efficacy with the same antigen that the vaccine is. And since outer surface proteins A and B are encoded on the same plasmid, they found a strain that didn't have the OspA and B plasmid in for their racketeering patent. But what Alan Steer did in Europe in this article, which we were not able to find out about until 2002, because it wasn't included in the Dearborn booklet. So what they did was they used high passage strains which drop plasmids. So if a person has an antibody against the gene product that's produced by that plasmid, you're not going to have a match in a Western blot. It's not going to show up. And the second thing he did was he used recombinant OspA and B with no lipid attached when it's the lipids that are immunogenic and not the proteins. So it's the lipids that are immunogenic. So this is how he comes up with wimpy strains that drop plasmids, don't have OspA and B in it, and more importantly, he, as you can see in the second graphic, he averages, and it was my friend Laura Hovind who goes by Borreliosis too, and who's a CEO and webmaster of Truth Cures. The lady can do everything. She's kind of like a jack of all trades. We had said that they had falsely elevated the lowest cutoff for the IgG ELISA by using a 5 standard deviation limit to exclude anything below that as baseline noise. But in fact, what they did was they averaged the IgG responses between meningitis and arthritis and erythema migrans and acrodermatitis, effectively excluding early and neurologic Lyme. Just by simply, you don't do that. That's not done. That's not how science is done. Typically, when you're developing an analytical method, you look for your method should detect the lowest possible concentration of the analyte in question, not the highest. So I just wanted to point out that it's not IDSA. People think IDSA is the criminal organization. It's not. They're not too bright a bunch. They're not really scientists. They haven't ever said to you that spirochetes are their own phylum, or shed fungal antigens. They've never said spirochetes are not regular bacteria. They don't have any lipopolysaccharide or LPS, and so they're not the culpable party here. It's the ALDF. So let's fast forward to the present era. Nicole Baumgarth publishes more recently in recent years that Lyme disease is a disease of immunosuppression, and do your own research on this. Look at her articles and follow up. In one of her articles from 2017, she gives an explanation of what B-cell germinal centers do, and she talks about how the premature collapse of B-cell germinal centers renders these immature B-cells that really are incompetent, and incompetent also to secondary infections like influenza. B-cell activation and proliferation in germinal centers is facilitated by interaction with CD4 T cells, and presentation of antigens on follicular dendritic cells. Despite the early presence of BB in the draining lymph nodes, germinal center B-cell and T cell numbers remain low for the first 2 weeks in experimentally infected mice. The germinal centers collapsed around 1 month after infection, and associated B and T cell numbers decreased steadily after that. So within the first month, I guess the majority of people who are going to go on to chronic illness are just going to just have see their immune system completely fold within the first month. Now, we can back up to 1988 and look at what Alan Steer and Paul Duray have to say. This article was found by Laurie Dennis in Canada. She's the lady who wrote that book, Lyme Madness, down the rabbit hole like an LSD trip into crazy land of Lyme. In it, you should read this in great detail, and I recommend everybody with Lyme have a copy of this with them whenever they go to any doctor, because look at the pathophysiology of this disease. Diffuse visceral involvement in its acute stage mimics infectious mononucleosis or disseminated viral syndromes. They go on to talk about tissue damage and organ damage. The plasma cell precursors are large, appear tumor-like, and can resemble Reed-Sternberg cells. Then in 1989, in its own journal reviews, there's an article by Andrew Packner who calls the neurologic manifestations of Lyme disease the new great imitator. And I point out that that's not the great arthritis only ator. Neurologic does not mean Dearborn case definition of arthritis only. And this next article by Alan Steer in 1991, I think I got this from Karen Forstner at the Real Lyme Disease Foundation, which was lyme.org. And it's Alan Steer talking about syphilis as now a competitor for the title of most complex infection because of the neurologic abnormalities it produces. Lyme disease is reminiscent of neurosyphilis. Okay, Alan Steer talking about a neurologic disease from tick bites, right? That's very different from what you hear today. And then he goes on to say, if Borrelia burgdorferi does cause this syndrome, it's absolutely amazing that this spirochete will mimic not only rheumatoid arthritis, but also multiple sclerosis, the two of the most puzzling and devastating autoimmune diseases. Now I would like to talk about the issue of seronegative Lyme disease. I am convinced this entity exists. Okay, so Lyme causes the diametrically opposed MS and lupus versus rheumatoid arthritis. Now, it's long been assumed or known that rheumatoid arthritis and MS don't occur together. But here he's saying Lyme can cause all these things. Well, that is a clue that maybe Lyme is activating pathogens that cause those two diverse, disparate outcomes. You might not know this, but there used to be a Lyme and lupus clinic at Yale because they knew that Lyme was associated with the production of lupus, and that the NIH had a MS and Lyme division under the National Institute of Neurological Disorders and Stroke. It was run by Roland Martin and Adriana Marquez. So let me suggest what might be the reason for MS or lupus or the Bell's palsy outcomes of Lyme. You can do your own research. And then in 2012, Jane Brody of the New York Times did a story on Lyme. She interviewed Adriana Marquez, who said, as you can see, there are other infectious organisms, Epstein-Barr, for example, that can produce similar symptoms and may be the real culprits. So we say, think. So now onward to the mechanisms of pathophysiology. Borrelia as shedders of fungal antigen. In this next slide, you see is entitled, Researchers Find Rewarding Careers as Software Entrepreneurs. But I don't think that's the correct title. I think that's a mistake. Nevertheless, this is in 1996 in The Scientist by Karen Hopkin. She quotes both Alan Barber and Steve Bartold, but Bartold goes on to say about the pathophysiology of Lyme that it's using some sort of stealth bomber mechanism or using another diversionary tactic called blebbing. The spirochete can pinch off bits of its membrane in order to release surface proteins. Explains Barber, it's like bacterial Star Wars, in which surface antigens might intercept incoming host antibodies. I'm proposing that maybe they're stealth bombing or flacking over the immune system with fungal antigens that render the immune cells unable to present antigen or become tolerant. So read this whole thing about how these shed blebs contain DNA by the NIH at the Rocky Mountains lab. Then they developed a detection method to detect the antigen, not the DNA, in these shed blebs, and patented it. So for the bad guys to say the urine antigen capture method by Igenex is bogus is false. The NIH were the ones who patented it in the first place. So they obviously think shedding or blebbing off of these antigens is a thing. This next graphic shows the comparison between the number of spirochetes versus the number of blebs. And there's a little arrow there to the bottom left that shows these ghost-like shed surface antigens. The NIH say they contain DNA, and we know they contain the surface antigens like the outer surface proteins. And that's probably what's causing the disease right there. So in 1994, one of the Lyme crooks are here. I see there's a couple. Dave Persing wrote about the target imbalance, about how they're finding more DNA from the blebs than the actual DNA from the organism, such as the flagellin DNA. And so they're wondering how that could be, and I'm proposing maybe it's shed in these blebs. Here's an article about how these circulating membrane blebs or vesicles or exosomes, whatever they're called, the terminology is still in flux. People are saying this, that, and the other thing. I don't think it's clear, but we'll call them blebs. These circulating bacterial membrane vesicles or blebs cause sepsis in rats. So then we find Diego Cadavid in 2010 saying these shed blebs from Borrelia can disseminate from the periphery to inflame the brain. I wasn't going to talk about this here, but I might as well since we just mentioned Adriana Marquez and Roland Martin. This is in 2006. The two of them published two articles showing that the Borrelia lipoproteins, the triacyl-lipoproteins of Borrelia, not necessarily OspA, cause immunosuppression. And in the next article, they admit that these antigens are TLR2 and TLR1 agonists, which means they're triacylated. And they go on to say that these lipoproteins cause immunosuppression in terms of the antibodies in the blood, but with chronic brain inflammation. Since the lipoproteins of Borrelia are TLR2 agonists, that means they're triacyl, and structure predicts function, and we can't recrystallize something that's highly lipidated and thus very endotoxic. We have to use biochemical means to see what toll-like receptors manage these shed Borrelia or other pathogens lipoproteins. And here we'll continue with Justin Radoff talking about, we observed that membrane lipoproteins and lipopeptides from Borrelia, treponema, and mycoplasma are managed by toll-like receptors 2 and 1. So we know that this is how these lipoproteins like OspA are managed. In the text of this report, he says, more recent observations suggest that the mechanisms underlying monocytic cell activation by motile, B. burgdorferi, and T. pallidum are identical to those employed by their purified membrane constituents. These results support the notion that lipoproteins are the principal component of intact spirochetes driving the host immune response in Lyme disease and syphilis. Similarly, lipoproteins and lipopeptides from the human pathogen Mycoplasma fermentans, blah, blah, blah. So it's the lipoproteins, and we're finding that this happens with other diseases. So that's the scientific method. You show how your mechanism occurs in parallel. And here's another report on Mycobacterium tuberculosis, a novel TLR2 ligand that inhibits MHC class II antigen presentation. So one of the mechanisms by which these Borrelia lipoproteins cause immunosuppression is to inhibit the presentation of antigen against which antibodies are made by the major histocompatibility complex. So you're going to have immunosuppression from these lipoproteins, and this is a mechanism right here. First, know that it's the lipoproteins, because they're triacylated, that do this. They're handled by toll-like receptors 2 and 1, and secondly, a mechanism, and there's more, by which they cause disease is to stop presenting antigen. And here we show it occurs again with Brucella abortus. Overall, these results suggest that the OMBs from B. abortus exert cellular effects that promote the internalization of these bacteria and downregulate the immune response. Another report, Brucella abortus downregulates MHC class 2, and it has to do with the membrane lipoproteins. But let's take a quick detour here and look at how it is known that Epstein-Barr also affects antigen presentation negatively. I think I have 3 reports here. The lytic cycle of EBV is associated with decreased expression of cell surface major histocompatibility complex 1 and 2. And the next one says downregulation of class 2 expression during Epstein-Barr virus reactivation. And the third one says Epstein-Barr virus evasion is concerted by multiple gene products, and they have viral, they have their own interleukin 10, which is another mechanism of immunosuppression. So there's 3 mechanisms, or 3 reports rather, that pretty much show you that reactivated Epstein-Barr can be seronegative. And that means that seronegative could apply to any condition that you have, and you could still have the chronic active disease. It also means that all the journal articles that show a negative association between the herpes viruses and post-sepsis syndrome have to be thrown out. You can't have an antibody association if they're actively inhibiting such a process. And then really that expands to the bigger picture. Anything in the journals that associates a disease process with antibodies has to be either thrown out or looked at again. And in this instance, we were proposing that perhaps the Epstein-Barr virus and the other herpes viruses are being reactivated, and that's how Lyme causes chronic disease. That you would have to have a monkey or mouse model where the mammal is co-infected with its own version of a herpes virus, and perhaps use some kind of radio-labeled Epstein-Barr to see if you have the same mechanism occurring. Are the Borrelia lipoproteins activating its own version of Epstein-Barr and other pathogens? Here we show that even in Staph aureus, which is a bearer of toll-like receptor 2 and 1 antigens, in parallel, it is the fungal triacyl-lipoproteins that are causing the disease. And MRSA obviously is fairly important. So we recommend everybody who's looking at this, all research, take a look at all the primary articles that discuss lipoproteins in pathogens and see what kind of bacteria use them or have them on them. And then you have to relook at, take another look at pathology associated with that. So now you have seen that highly acylated lipoproteins from other organisms, Mycoplasma, Treponema, Mycobacteria, Brucella, Staph aureus, also cause immunosuppression. So that's how you do science. You have to show that your mechanism occurs in parallel, in an independent model. You get the same result. And this is OspA, 1,2,3 triacyl-lipopeptide. It is these acyl groups that cause the endotoxic response and not the protein end. And I wanted you to notice that in 2012 in the New York Times, they talk about in the 1930s, they invented thimerosal to prevent fungal contamination in multi-use vials. So that means thimerosal was literally invented to prevent Limerick's, if you can believe how stupid this is that we're dealing with. This is a report from 1953 where they co-infected a type of mouse with both Mycoplasma, also called erythrozoa, take a clue from that, and a hepatitis virus, and found that it actually killed the mice. So in combination, you have this immunosuppression from fungal antigens with viruses, and you can get a deadly outcome. So that's probably the reason why thimerosal was invented, to prevent the likes of Limerick's, a fungal endotoxin. So use your Google machines and your PubMed machines, because doctors don't. Here are the results of the scoring of different undergraduate majors with the MCAT, or the test you have to take to get into medical school. And we find that doctors, in fact, or people who had pre-med or biology majors scored the lowest. And they say, of course, there are a lot of things unsaid here, and it would be pretty illicit to say that the sole determining factor between these groups was their choice of major. One could easily make the argument that a certain degree of selection bias exists here, but it's difficult to quantify. Personally, I suspect that the primary reason for the discrepancy between the biology and the physics and engineering majors types is that the MCAT is designed to test problem solving rather than rote memorization. It shouldn't surprise anyone that students who spent 4 years learning to solve problems outperform students who spent 4 years memorizing their lecture notes. Or it just could be arrogance, in my view. I call it the Dunning-Kruger syndrome. I felt like it was cops and doctors are like cults because they protect each other first. They don't think outside the box, and they just only recite the cult's dicta. Greed, arrogance, pride, avarice, pride being the source of all offenses to God. And I am the Lord thy God, thou shalt not have self-EMDs before me. So use your Google machines or your PubMed machines to look up the meanings of tolerance and cross-tolerance, as well as see that, quote, dormant viruses reemerge during lingering sepsis. You'll be able to see that, in effect, they mean the same thing, and that post-Lyme syndrome or chronic fatigue syndrome are forms of AIDS or acquired immune deficiencies that are called post-sepsis syndrome. So go ahead and do that. Use your Google machine. And then let's just use this description of endotoxin tolerance by Andrei Medvedev as a definition, and we'll call ours fungal endotoxin tolerance to show that TLR2/1 agonists, or the triacyl-lipopeptides from Borrelia, mycoplasma, or whatever, are fungal endotoxins. This is a description of post-sepsis syndrome. Disabling muscle and joint pains, extreme fatigue, poor concentration, decreased mental functioning, and so on. So if anybody who has chronic fatigue syndrome or Lyme or Gulf War illness is reading this, you'll certainly identify with these as the primary symptoms, maybe not in the order listed here. But the net effect of tolerance and cross-tolerance from borreliosis is the same as post-sepsis syndrome, chronic active viral infections and other kinds of infections, but no immune response. Next, you'll see how tolerance to toll-like receptor 2 agonists cause cross-tolerance to other toll-like receptor agonists, such as 4 and 7/9, which deal with lipopolysaccharide and viruses, and to flagellin, which is a toll-like receptor 5 agonist. The great imitator was probably all along Epstein-Barr and friends, and the latent viruses are activated by immunosuppression. So here's Nicole Baumgarth in 2015 talking about how she co-infected mice with an influenza virus and found that those mice could not fight off that secondary infection. So this is the model that we have been showing you in practice in an in vivo study. In 2003, Hulinska et al. Did an experiment in vitro with spirochetes and the Epstein-Barr virus, and found that, yes, spirochetes help reactivate Epstein-Barr. You can do your own research, use Borrelia or TLR2, and see if it's well known that fungal antigens of this type induce the immunosuppressive cytokine interleukin 10. Yes, Mario Philippe showed that, I think this says 1998, I can't really read it, that in fact OspA induces the immunosuppressive cytokine interleukin 10. Other examples of fungal antigens inhibiting apoptosis is Mycobacterium bovis, BCG, inhibits apoptosis. Mycoplasmal infections inhibit apoptosis and induce malignant transformation. Mycoplasma tuberculosis inhibits immunity by inhibiting apoptosis. Candida albicans, the same thing. Mycoplasma fermentans, the same thing. And they're saying that the source of this fungal antigen-induced inhibition of apoptosis was the lipoproteins. So we're showing this again in parallel, another mechanism in parallel. You can go ahead yourselves and try this experiment to see if fungal antigens inhibit apoptosis or cause immunosuppression. There's a search strategy right there. Another one, use this language, acquired immunoparalysis or immune paralysis acquired. Interleukin 10, and whether or not that downregulates the antigen-presenting cells or inhibits antibody production. And here is TLR2 agonists causing brain damage. So OspA might not be good. Fungal contamination of live viral vaccines might not be good for children. And so now you've seen that Limerick's and Lyme and the antigens like it, which is required of the scientific method. You have to show your model occurs independently. Inhibit antigen presentation, induce the immunosuppressive cytokine interleukin 10, inhibit apoptosis of possibly latently infected immune cells, i.e. EBV, are associated with tolerance and cross-tolerance to other pathogens that bear LPS, viral, and TLR5 agonists. And finally, chronic active viral infections can be antibody negative. So any associations in the literature have to either be thrown out or looked at again. Next, you're going to see who else says OspA was the opposite of a vaccine. Dave Persing, Robert Schoen, the NIH, you already saw that, Anthony Fauci, Lyndon Hu, Alan Steer, Raymond Atweiler, Gary Wormser, Paul Mead, Donald Marks, and even GlaxoSmithKline themselves. So this is a patent which I call the RICO within the RICO patent. It was applied for by Dave Persing, but it was worked on by Robert Schoen too. And in it, they claim that you can't tell the difference between Lyme and Limerick's vaccination, and that it has to do with the multi-system complaints characteristics of later presentation of Lyme, which means he's not talking about exclusively a bad knee. So here in the patent, they claim they can't tell the difference between Lyme and Limerick's disease, and that they're multi-system or post-sepsis. This patent was applied for before they filed this report that goes with it. So you see all the names of people that were involved in developing this method, where they use a strain of Borrelia burgdorferi that's missing the OspA and B plasmid. Ben Luft at the FDA, there's the link. It was originally stated that when we got the overview of the disease, that the disease was really quite protean, that means multi-system, and actually the adverse events are very similar to what the disease manifestations are. So Ben Luft is telling the FDA, you can't tell the difference between Lyme and Limerick's disease, and it's not just a bad knee, it's multi-system. Here, Donald Marks, who was an OspA vaccine trial administrator, went down to the FDA in January of 2002 and said, he's quoting the SmithKline Beecham and saying, we can't tell the difference between the natural history of Lyme disease and vaccine injury or failure. As you saw this previously, Adriana Marquez and Roland Martin, 2006, say the same thing. OspA and antigens like it do not produce arthritis only, they produce systemic immunosuppression with chronic brain inflammation. And most recently at all, and the reason I even decided to make this video is we found this grant applied for by Lyndon Hu, who, as you saw, was a co-author of the nonsense report about long-term treatment with Mark Klempner, based on the Dearborn case definition. And here Lyndon Hu is saying something that's the exact opposite. He sounds like this grant application sounds exactly like if we, the Truth Cures team, was writing it. And he's talking about the anti-inflammatory effects of TLR2 agonists or Limerick's disease, causing tolerance, mutualism, or benign neglect. And then he goes on to say, we believe that the development of tolerance to repeated stimulation by organisms that cause long-term infection, that a diminished immune response is an important mechanism for self-preservation in infectious diseases such as Lyme. Well, that's the exact same things Medvedev, of course, and others have been saying. But it's different to hear it, in Lyme land, where we have been for the past 24 years fighting against this ridiculous case definition where you're not allowed to have Lyme disease unless you're not sick and it only affects your knee, but get the vaccine. As I said, this is who Lyndon Who is. Excuse me. Anthony Fauci, director of NIAID, the National Institute of Allergy and Infectious Diseases, has a patent that says other diseases characterized by a decrease of T-cell immunity, for example, mycobacterial infections like TB and fungal infections like cryptococcal disease, this treatment will be good for, and that it can be also used for the treatment of secondary infections that occur in patients with suppressed immune systems, such as the opportunistic infections that occur in AIDS. Sorry, so here's Anthony Fauci saying fungal infections cause immunosuppression and the reactivation of latent viruses, etc. Here, Gary Wormser and Timothy Stellati in 2012, talking about basically how the fungal-ish OSPs like OspA cause immunosuppression, which means the Dearborn case definition is bogus. In 2016, look at this list of characters. Alan Steer, Gary Wormser, Lyndon Who, Paul Mead, and there are more, I think, familiar names. And they're basically saying also that Lyme and Limerick's are about immunosuppression, and they cite Mario Philippe saying OspA causes the induction of the anti-inflammatory cytokine interleukin 10. The anti-inflammatory effects may be the more important function of TLR signaling. Right, so Lyme is not about a single solitary oligoarthritis in a knee where the patients feel fine except for their arthritis symptoms. So you know that this whole definition of Lyme, this whole scam is a lie, because now all these people, we really can't apply for grants having anything to do with Lyme unless they're talking about TLR2 agonist tolerance. Here Gary Wormser said it in the year 2000. The OspA vaccine for dogs seems to cause immunosuppression. We don't know why. And I'm saying, well, it's the fatty acids, because you have to know what stuff is to know what it does. And this is Alan Steer in 1990 using the seronegative Lyme assay. Later he said there was no such thing as seronegative Lyme, that those patients were psychiatric. In the methods he shows, this is a statement, if the patient was seronegative according to these methods, the serum was further tested by immunoblotting, and peripheral blood mononuclear cells were tested for reactivity with Borrelia antigens by the proliferative assay. That's Datwyler and Volkmann's assay from 1988. Before we start looking at the proliferative assay, I want to show another report that came out at the same time by Datwyler and Volkmann, modulation of natural killer cell activity by BB. And in it they talk about how the inhibition of natural killer cell activity also occurred as a result of exposure to the supernatant, where it says at the bottom there, the inhibition is directly attributable to the organism or its products, and supernatants from the organism's cultures also inhibit endogenous natural killer cell activity without prior exposure, data not shown. So that's important because it shows that it's the Borrelia lipoproteins that inhibit natural killer cell activity. That's another mechanism. I don't know whether it's related to the previous ones that we've seen or not, but there it is. So at the same time, in 1988, Datwyler and Volkmann and Halperin, Ben Luft, Go Lightly, published a seronegative Lyme assay. And the whole purpose of this, of course, is to discover if patients who are antibody negative, who still have chronic symptoms, were antibody positive in the past, perhaps. Could you tell whether or not it was Lyme that they had been exposed to that causes chronic illness? And, of course, they developed this assay. So the more serious and disabling form of Lyme is the seronegative kind, chronic neurologic, or the triad. And Raymond Datwyler even said this in front of the FDA committee in '94, before the Dearborn event happened. He says the sickest patients are seronegative. Now, Alan Steer had used this seronegative Lyme assay to look at chronic neurologic Lyme patients in 1990. And so subsequent to that, in '94, before Dearborn happened, Raymond Datwyler was talking about this. These are the FDA 1994 FDA vaccine meeting minutes, and he says that the patients with a poor immune response tend to have worse disease. That's page 49. The very next page, Alan Steer is in the room. You remember the old Nazi trick? I think it was Albert Speer who said, I walked out of the room during that point of the meeting. Well, Alan Steer was here to hear Raymond Datweiler say the seronegative patients were the sickest. And here you see Alan Steer talking about it's like syphilis and blah, blah, blah. But anyway, I wanted to show you that these are the meeting minutes from 94. Because subsequent to this, 5 months later, 4 months later, whatever it was, in October of 1994, they held the Dearborn stunt, and they excluded all these seronegative sickest patients and made Lyme to be just about late Lyme arthritis with no other illness signs. And those are just the meeting minutes. Let me see, where are we? Oh, continuing with the libel. This happened in 1993. Alan Barber and Derlin Fish wrote this article slandering, libeling, all the Lyme victims and heavily misogynistic statements. But the important part of this is that they mention that the early phase 1 and phase 2 trials of the OspA vaccines, one of which was owned by Alan Barber and the other one was owned by Yale, almost like preemptively guarding against these chronic late neurologic post-sepsis Lyme outcomes, because they knew in 93 that they were caused by the vaccine too. Hence, 1994, the Dearborn stunt to exclude them from the case definition. Once again, you saw this before. Laurie Dennis found this in Canada. We had 2 other examples of Paul DeRay saying that Lyme results in what looked like Epstein-Barr transformed cells and premature-looking, immature-looking lymphocytes in the spinal fluid of Lyme victims. I'm just bringing this up again to show that there's a couple more. And what is the definition of this Reed-Sternberg cells? You can Google that. There's a heavy association to Epstein-Barr. So that's what we're talking about. It seems that empirically, the pathophysiology of Lyme disease seems to be about post-sepsis, immunosuppression, the reactivation of latent herpes viruses, and there it is. So this excerpt where it says Paul DeRay on the top left is from the 1992 book, Lyme Disease, Molecular and Immunologic Approaches, which is a conference summary that which took place in 1992 at Cold Spring Harbor. And Paul DeRay once again says these immunoblastoid cells in BB infections at times resemble those found in Epstein-Barr virus infections. Does BB reactivate latent virus infections in tissue? Do some tick inocula deliver simultaneous infectious agents that produce multi-agent infections? Well, you almost hit it. And of course, there still could be other pathogens in a tick, spiroplasma, mycoplasma, that have never been detected and discussed because, why would we? First, we have to have the Limerick's roll out, and then all the blood and potential patentable antigens in them have to go to Yale first, and Crooks and Immugen. So that's the book I got this from. And then the third time, Paul DeRay said that these look like Epstein-Barr transformed cells in the chronic neurologic Lyme patients was in It's His Own journal. They used to have a journal called Reviews of Infectious Diseases, and they had a special supplement come out in September, October 1989, just on spirochetal diseases and Lyme. And importantly, this is the third time DeRay says this. Immature B cells can also be seen in the spinal fluid. These cells appear quite atypical, not unlike those of transformed or neoplastic lymphocytes. Of course, we know the Epstein-Barr immortalized cells could be considered neoplastic. Whether or not there's a correlation between Lyme disease and tick bite sepsis and cancer, it probably exists, but we're not allowed to know about it, just like we're not allowed to know about the long-term outcomes of children who are born with congenital Lyme or congenital Epstein-Barr, cytomegalovirus, and so forth. That's really, I think, what's driving the disease here. And finally, here's OspA speaking for itself. It says that fungal antigens inhibit B cell maturation, which is what DeRay and Steer had been talking about all those years. So now, who have you seen say OspA, panthericys? Panthericys is an analog of OspA. OspA is a triacyl-lipoprotein. It behaves like. We don't know if it's exactly like panthericys, but OspA and the fungal lipopeptides of Borrelia behave like that. So who says so? Dave Persing, Robert Schoen, Ray Datweiler, Alan Steer, In 1990, Gary Wormser, CDC's own Paul Mead, Paul DeRay with Alan Steer, Donald Marks, GlaxoSmithKline, Ben Luft at the FDA. Donald Marks himself put out an article talking about the neurologic outcomes of OspA vaccination. Remember, we're not allowed to have that. Lyme disease, quote unquote, means only a bad knee. Anthony Fauci, who's the director of NIH, NIH's own Adriana Marquez and Roland Martin, and finally Lyndon Hu. So except for Ben Luft and Donald Marks, those same people, the cabal, Datweiler's iffy. He's made some derogatory statements about Lyme victims, but for the most part, we rely heavily on his Lyme is a seronegative disease science. So those people who falsified the case definition clearly did so to pass off this bogus vaccine, Lymerix. They knew that they were going to injure these people, so they said, well, let's just exclude that systemic post-sepsis syndrome outcome that comes from jabbing people with a fungal endotoxin. And everyone should know that their behavior by issuing guidelines, every time they make a statement about Lyme, that is based on the Dearborn case definition, basically resets the clock on the fraud case, but also what they're doing is maintaining the pretense, that's a legal word, that the Dearborn case definition was real. This is what they're doing if you watch closely. Once again, this is the case definition. You should burn this into your brains. This is how they falsified the case definition. They left out Neurolyme because it was caused by OspA. This was filed with the Justice Department in 2003. And my friend Bo, Laura Hoven, made this movie, which you should go and look at if you're a novice to this. But now I'd like to move on and discuss some of the other Nazis that we have to deal with. The first one is Suzanne Vernon. She's a CDC employee. She published this report called The Absence of Mycoplasma Species DNA in Chronic Fatigue Syndrome. Research fraud. Here we show that she has thrown out or centrifuged out the mycoplasma before looking for it. And this next image, it shows these erythrozoa or mycoplasma adhering to red blood cells. They depolarize the red blood cell membrane such that it can't transfer oxygen across it. So most certainly they are involved in post-sepsis syndrome, particularly when it comes from a fungal disease, one that you can't fight off because you're now tolerant to TLR2 agonists. And go ahead and look that up for yourselves. Here are some of the other names for mycoplasma and whether or not they adhere to erythrocytes. Of course, that's what they do. And they're also intracellular, and they adhere to mitochondrial membranes, also inhibiting mitochondrial function. Another character, this is Paul Auerwater. He's a funny little man, has some funny things to say. But in the criminal charge sheets on the Truth Cure's website, number 6, this is this is Paul Auerwater talking about whether or not he finds active vaccine strains from the MMR in children that have progressive vaccine-induced disease. And then he goes on to say, however, fatal infections have been documented in immunodeficient children vaccinated with these strains. She lists 4 references. And basically he's saying that Andrew Wakefield was right. It is the live viral vaccines that are causing this brain damage that we call autism, and I have a sneaking suspicion that that's not the language the CDC uses. They don't call vaccine brain damage autism. They think that's something else. They call it vaccine-induced brain damage. So there is more scientific data published on this. It's in our criminal charge sheets, but the CDC and Big Pharma don't want you to know that a mechanism of illness is this immunosuppression and the reactivation of viruses. You can see that this all overlaps: chronic fatigue syndrome, Lyme, and the brain-damaging vaccines. You're giving children live viral vaccines, and they are just not able to handle them. And it's not because of any other reason. It's not because of thimerosal. Thimerosal prevents the growth of fungal antigens. So this is the main page of the criminal charge sheet number 6. There's the web address in red. You can peruse that. For more of this evidence, you'll find IDSA's president, Amanda Jezik, saying at one point, at this age, infants are at greatest risk for certain serious medical adverse events, including high fever, seizures. The outcome of the seizure seems to be what the CDC is alluding to when they're talking about long-term damage, and sudden infant death syndrome. Therefore, it is important for the ACIP to consider carefully the risks versus the benefits before making a recommendation, rather than to be on a forced schedule that suits the manufacturer as opposed to the patient. So that's been taken off the web, but we've screen captured the image and the statement. So parents should know, just as there is a semantics game going on with Lyme disease, so there is with autism. So look at their website. There's the address. They say, tell your vaccine provider if the person getting the vaccine is immunosuppressed or is not feeling well. Does that ever happen? No, and I don't even think doctors know. You're not supposed to vaccinate kids who are sick. So a child can become brain damaged from the live viral vaccines if they're immunosuppressed. They have some other infection like a cold going on, fever, or the vaccine vial is contaminated. So there it is. It's the same model. And then there's Paul Offit. Now, I think it's important to look at this guy because look at this face. This is a violent reaction to something he is in fear of. And the essence of cowardice and then aggression is the fear of being less than someone else. And the last thing you'd ever want to be is a coward. Human nature despises a man with no guts, worldwide and throughout history. And here's another our waterism. There was a video article that he did on Medscape, and the title of it was Candida auris, time to prick up your ears? Hello, this is Paul Auerwater, blah blah. Candida and antifungal resistance, to me, as a non-mycologist, seems relatively static. There's the link. Just say, I'm no mycologist, and then you're off the hook, and that's your get-out-of-jail-free card. Another character, the infamous Simon Wesley, the things that he's done. You may not know this, but, and this is in the criminal charge sheet, we have a special one just for him. The role of vaccinations as risk factors for ill health in veterans of the Gulf War. Of course, he says there is a specific association between multiple vaccinations given during deployment and later ill health. So he did find for the Pentagon that there was a relationship between vaccines and Gulf War illness. Later, at a public speech in 2006 called Something Old, Something New, Something Very Much Smells Like Bullshit, he says one way of doing, in terms of assessing Gulf War illness to see if they have valid illness signs, one way of doing that is through neuroimaging, but we didn't get the money to do that. So instead we use sophisticated neuropsychological checklists. Right. And then when he's discussing the immunosuppressing DEET exposures or nerve agent antidote, he says, well, we don't think that was an issue for the British Armed Forces. But we do think there is a particular pattern of protection in what happened later. So in other words, he's saying nothing's happened. There's no real illness. They're just psychiatric, and they're just scared. They're scary fairies, soldiers, and that's why they're not really sick. And Wesley was proven wrong about chronic fatigue syndrome and Gulf War illness. As you can see, antibody wipeout found to relieve chronic fatigue syndrome, and Wesley's explanation for his lifetime of criminally stupid behavior is to say that the belief that chronic fatigue syndrome is all in the head has been around since the beginning. In other words, misogyny's been around since the beginning. So that makes it right. And that was his excuse. He didn't have an excuse. Well, I call that Dunning-Kruger syndrome. Wesley had written an essay on pride, basically saying it has to be earned. But one of the symptoms of Dunning-Kruger, you can use an anagram DKS, go ahead and say that, is being unaware of your mental performance and thus being in denial of your capacity to think outside the box, which perfectly fits Wesley in my view. So remember, the MDs score the lowest on the MCATs too, so you could call that the MDs score lowest on the MCAT syndrome if you like. But I think people should know about that there are parallels in these other areas, Gulf War illness, vaccines, childhood vaccines, and the brain damage. as well as Gulf War illness. It's research fraud and slander and libel. That's how we get treated. But this might be the most outrageous of all Lyme lies. That humans can disinfect ticks with OspA antibodies. I have a special criminal charge sheet on that, and you can read that that's all research fraud. But let's look at the main claim that they make for how Lymerix works, or the recombinant OspA vaccines. Thus, the vaccine blocks transmission in the tick, and the spirochetes never enter the host, which is false. So the only claim about the recombinant OspA vaccines allegedly prevent Lyme was to say that the humans could become walking canisters of tick disinfectant. What's next? Should we just be all swallow Roundup and go pee on the weeds? How ridiculous. Nobody's going to go for that. This is a completely bogus article because what they did was not do any kind of xenodiagnosis. They simply used immunoradiolabeling to see if an OspA vaccinated mammal, when a tick sucked on it, that there could be any remaining spirochetes in them. But OspA is not expressed at 37 degrees C. OspC is, and they didn't use any kind of a valid method to assess its efficacy. So this claim is entirely false. And lastly, and this may be even funnier, the two vaccine patent holders, Yale's Errol Fickrig and Richard Flavell and Alan Barber, who owns Immulyme, both published that these vaccines would not prevent Lyme due to antigenic variation or, quote, selection pressure, which means you cannot have a vaccine for relapsing fever ever. Never mind OspA. And here's the first report on that. That's the name of it, selection of variant Borrelia burgdorferi isolates, blah, blah, blah. They don't work. And Alan Barber in 1992 says that OspA undergoes true antigenic variation. He later said it didn't. But here we caught him in another lie.